Okay, as people take their seats, we can start to get into the next session for the meeting. As you've seen, this is bringing together people from different kinds of backgrounds, and the diversity of backgrounds will become even more apparent as we go into the next few talks. One of the things that's actually quite exciting about this whole effort is the extent to which it has brought in people from very different areas and now are thinking hard about these problems, as well as working with people who have been already grappling with the neuroscience aspects of these problems. So it's my real pleasure to welcome Jim Schuck to give our next talk from the Lawrence Berkeley National Laboratory, from the molecular foundry there, sort of their nanoscience haven. And he will speak to us about upconverting nanocrystals for deep brain probing.
Ralph, thank you for the invitation. And thank you for the opportunity to come down and participate in this symposium. I've already filled up pages with notes and questions, and hopefully, it's later on you know that I'm not copying your work. I'm trying to call you and ask you about it. All right. So actually, before I get started, I'd just like to point out, the pretty images, or at least pretty to me, images that are on the program and on your name tags and everything, are actually somewhat abstract ball and stick models of up-converting nanoparticles, sort of at the atomic scale. Each color is a different element. They were made by a person who is now an undergraduate here at UCSD, Andrew Mueller. So Andrew actually made these when he was a high school student working in my lab for a summer. But if any of the local folks are interested in finding someone to do some graphic design for the next cover image or anything like that, I'm sure for a price, Andrew's available. Anyway. All right, so we heard in our the first talk about fluorescent proteins and small molecules that emit light. Next talk we heard about molecular probes for magnetic resonance imaging. I'm now going to keep going, and I'm going to talk about nanoparticles and maybe their applications or potential applications for nervous system probing. So in my title here, you see that I put a question mark after deep brain probing. The reason for that is because we, and actually really no one I know, has been very successful in putting these types of nanocrystals, the upconverting nanoparticles, really into brains and using them for imaging. So if you're here waiting for the in vivo data, you can take an early lunch, I guess. But we do think they're promising, and I'm going to tell you why. So as Ralph mentioned, I work at a place called the Molecular Foundry. The Molecular Foundry is a national nanoscience research center. We're housed at Lawrence Berkeley National Lab. The idea is that nanoscience really is a very inherently multidisciplinary sort of field. And so this building really was a big experiment by the DOE. They built the building and threw a bunch of people that have very different backgrounds into it, but all sort of nominally work at nanometer length scales, and hoped we'd talk. And I have to say, actually, it's been more successful than I would have guessed. As I mentioned earlier this morning to someone, it's part of that success is due to the fact that there's a big coffee machine sitting right here, so we all tend to run into each other a lot. But it is very good to get people to talk to one another. It very much is a collaborative environment, and so all the work I'm going to talk about here is very much done within sort of the confines of collaboration. The building itself is boldly cantilevered over the Hayward Fault up in Berkeley. It was designed this way to, I guess, sort of at least in an abstract sense, resemble one of the main tools of nanoscience, the AFM atomic force microscope cantilever. And so that's what this is here hanging out. You probably noticed that there's not a tip hanging off of the cantilever. It does have an oscillating frequency, about 3 hertz. Actually, a few years ago on April 1st, though, some of my postdocs snuck into our offices up here and hung a tip off of the cantilever. So then we felt like the building was complete. You can actually see we put a little optical antenna at the end of this tip as well, and we caught the sun reflecting off of it, so we thought that image was kind of cool. It doesn't necessarily relate to what I'm going to talk about today, but it's kind of neat stuff. Okay, so let me first just kind of tell you about the general theme of what goes on in my lab. So I would say what we think about are probing local environments with light. That's sort of what we do. One half of my lab, I would say, is dedicated to using the concepts of optical antennas and plasmonics for grabbing light and squeezing it and concentrating it down to nanoscale volumes, zeptoliter volumes, if you will, and then using that, well, so controlling light and then using it to do advanced near-field spectroscopic imaging. I wonder if I can make one of these work, maybe not. Let's see. No, it doesn't want to play. Anyway, we also tend to, you can't see here, but these are grapes in a microwave. Turns out antenna concepts work at all electromagnetic frequencies, and you can make little fireballs with grapes at microwaves, if you like, using very much the concepts of antenna theory. The other part of what we do is we try to use luminescent particles and luminescent molecules too as reporters of local environments. And in particular, we've spent the last few years concentrating on a type of particle called upconverting nanoparticles. And so that's what I'm going to talk about for the rest of today. So quick outline to the talk. I'm going to tell you about this relatively new class of nanoparticle, these UCNPs. In particular, I'm going to tell you about how we've been developing them so that they might actually be useful in sort of some of these contexts. And in particular, we've kind of undertook a photophysics investigation of these particles and realized that essentially everyone was making them wrong. And so we're trying to now optimize them for real applications. And then I'm going to finish telling you why we think they really could be interesting for these types of applications that we're hearing today. Okay, so people, of course, have been using fluorescence to detect single biomolecules for more than 50 years now. This is a paper that was I've turned on to by Steve Boxer at Stanford. Since this paper came out in the last 50 some odd years, of course, there's been tremendous progress in looking at fluorescent molecules and using it to study biology. People now, of course, can very easily track single, well, I would say we're at the point now where single molecule imaging is commonplace. We've gotten really good at it. You can now track single molecules moving around. You can use them to do super resolution inside of live cells. The reason for this progress really has had a lot to do, I'd say, with advances in a number of other technologies. So things like lasers, in particular optical filters. Of course, advances in computers and computing technologies that allow us to take lots of images and analyze them. And then also advances in detectors and CCD chips that let us detect single photons. What I would also argue is that relative to these other technologies, there's been very little progress in picking out probes. Now, I say that partly, of course, to be provocative in this audience. Of course, things like fluorescent proteins and quantum dots have made a huge impact on bioimaging. But what I really mean is sort of the approach of finding sort of what your best probes are hasn't really changed in the last 50-some years. So that approach, sort of in a nutshell, right, is you take your emitters and you throw them, I don't know, in a cuvette, and you shine a laser on them, and you see if they emit, and the ones that seem to be the brightest in this ensemble, then you start to optimize them. And so you test them for stability, and once you find ones you like, then you start pulling them out and tweaking them a little bit, so increasing their quantum efficiency by, I don't know, making a molecule more stiff, or increasing their optical cross-section by adding another conjugated bond or something, change their color too. But this has basically been how it's done, and this is actually how people have screened for the brightest upconverting nanoparticles as well. So let me tell you what these are. So an upconverting nanoparticle is essentially a ceramic nanoparticle, in our case sodium yttrium fluoride, that is doped with lanthanides. And what they do is you shine near-infrared light on the particles, and they emit anti-Stokes shifted light, so visible light in this case. By far, what had been determined to be the brightest and the most widely used upconverting nanoparticle are these sodium yttrium fluorides. Actually, people use different host matrices, but ones that are doped with very specific concentrations of erbium and ytterbium. And so erbium and ytterbium do a good job of absorbing 980 nanometer light and shooting out red and green light out the other side. You can see in this picture down here, really the particles look essentially transparent to the visible, but if you're shining infrared light through it and you turn the lights off, you see that there's a fair amount of green light coming in. So this was shining a watt of 980 light through a cuvette. Don't tell the laser safety guys, but it makes for a cool picture. The way so that these work, essentially, is that your ytterbium acts as sort of a molecular or an atomic antenna. They have much stronger absorption cross-sections than a lot of the other rare earths. So it absorbs the 980 nanometer light, and then it transfers that energy over to the other, to other lanthanides, in this case, for example, erbium, where it transfers 1 photon's worth of energy over, then it can absorb another photon and transfer it again. And that allows you to work your way up this excited state manifold and emit in the visible. So it's a multi-photon process. And you can begin to ask, well, how does this compare to the probability of multi-photon? How does this compare to 2-photon excitation in other types of particles? And so if you look at, say, an organic fluorophore, something like a rhodamine, you can ask how much, what's the peak excitation power you need for getting a 2-photon emission process to occur, so that you can actually detect the fluorescence coming out of the particle. And you see that power is very high, right? Something like 10 to the 10 watts per square centimeter is needed to really get a measurable 2-photon signal. You can use a quantum dot for doing the same thing. And again, the powers that you need are very high. It turns out that for the upconverting nanoparticles, you need much less power for getting this upconverted light. And the reason for that is actually very simple. So in most 2-photon fluorescence that people are used to thinking about, what you're doing is first exciting your molecule up to a virtual level, and then you have to excite again to a real excited state. Here, our intermediate states are real. They're real metastable states, and we excite to them first, and then while they're hanging out in this real state, we can excite up again. And so that's why they're so much more efficient. So what's the catch? The reason these that work so well is because it turns out that a lot of these states have very long lifetimes. They're not fluorescent, they're phosphorescent for the most part. And so they have lifetimes on the order of hundreds of microseconds to even milliseconds. So back in 2009, we showed that these things actually are great for single molecule or single particle level imaging. So here's a bunch of single upconverting nanoparticles that you can see quite nicely. We had to prove that they're single molecule or single particles, so we took electron microscope images of them. Another great thing about these particles and why we think of them as maybe near ideal single molecule probes is that they don't photobleach. Right? So here is a emission time trace where we turned on the microscope, went to lunch, came back, and of course it's still emitting at the same brightness. We've now been studying these things for 5 or 6 years. We've not seen one photobleach ever. The reason, of course, is because in, say, a 20 or 30 nanometer nanoparticle, you have a few hundred to a few thousand emitters. So really what you're seeing is emission from lots of different centers all at once. So that's what's going on here. It's not like a quantum dot where you have a single emitter that's then turning over one after the other. They also don't blink, turns out, for much the same reason. And so this is great, really, right? There maybe are, in many senses, ideal for doing single molecule imaging and as fluorescent probes, or as luminescent probes, I should say, right? You get background-free excitation, so you get to excite with the IR and look at higher energies. Another great thing is because they don't photobleach and because they're very photostable, they're actually, you can collect as many photons as you want from any given particle, right? So for a good fluorescent dye, you get maybe 1 million photons out of it before it dies. In this case, you can collect signal forever, and that allows you to get very high, you're able to localize the position of that particle very, very well if you're willing to wait long enough to collect those photons. All right, so they're great in many ways. We put them into cells a while ago, and in fact, so the community really is catching on, and so at this point, people are taking these particles and putting them in lots of biosystems, and in fact, you can find a fair number of review articles on these particles being used for doing bioimaging. And again, for the most part, these particles are the sodium yttrium fluoride, erbium, ytterbium-based particles, or some small variants of those, but that's pretty much what's used. And so, of course, we realized that there was still a lot of room for optimization and improving these if we really wanted them to be useful. So, of course, one of the things that you would like to do is take these particles and find a way to make them as small as possible. I mean, there are plenty of bioimaging applications where a 30 nanometer particle is fine, is great. But maybe if you want to use it to stick to something smaller and watch it move around, you don't want it to be heavy cargo, right, as it drags around. So you'd like it to be small if possible. you'd like these things to be able to emit in many colors. Turns out that's actually relatively easy to do simply by changing the lanthanides inside of the particles. And then, of course, you want them to be as bright as possible. That's true for any sort of luminescent type probe. A couple years ago now, Bruce Cohen set out to figure out how to make these things not 30 nanometers, but how to make them small, and came up with a great way to do that and show that he can now make sort of routinely sub-10 nanometer particles. This is what they look like. As you might guess, we'd take these down to our lab, though, and if the fluorescence in these, right, I keep saying fluorescence, the luminescence in these, the phosphorescence, really should scale, because it's just based on the total number of emitters you have in your particle. It doesn't scale very favorably with volume. And so as you take these particles down to very small sizes, they get very hard to image. And so that was the first thing we saw, and we basically decided, well, how can we make this better? So we realized we had to understand how these things work, and we undertook this whole study to understand the photophysics inside these particles by looking at the kinetics, and looking at the lifetimes, and also studying how things changed as a function of size. So we actually understood what the bottlenecks were in terms of energy transfer and ultimately luminescence. So, of course, one of the first things we noticed is that when you have pretty big particles, the way that the luminescence scales with volume is exactly what you expect. As the volume gets smaller, the luminescence goes down linearly with that relationship. But once you get down to about 20 nanometers, you actually start to see that it gets even worse, that the brightness really starts falling off even worse than you'd expect for this ideal volumetric scaling. And in fact, what you see is that at about 10 nanometers, 8 nanometers, more than 75% worse than you would have expected, right? A factor of 4 or less. And it turns out that really what this means is it's effectively like having a dead surface layer of your nanoparticle that's a couple nanometers in size. So surfaces are clearly having an effect, right? In fact, we would argue that these particles, you can't really call them nanoparticles until surfaces start to dominate, right? And so this happens right at about 20 nanometers or so. It's when they're truly nanoparticles. Something else we start to notice with these small particles, again, that are made of primarily 20% ytterbium and 2% erbium, is that actually there's some spectral heterogeneity as you go from particle to particle, which had never been seen before. Again, if you have a particle that has hundreds or thousands of emitters inside it, even at 10 nanometers you have 100 emitters inside these particles, you really wouldn't expect to see much spectral change, at least within some sort of Poissonian distribution. But what we see is that for some very specific lines in the emission, sometimes that peak is there or not there. Sometimes it's there and it's the brightest transition in the particle. So again, clearly something's happening as you get these things smaller. Actually, we don't have a good explanation for that heterogeneity. But what we do know is that it goes away when you put a passivating surface layer on top of it. So clearly it's surface related. We used to think that maybe this was proof that actually only have a couple of emitters that are working at any given moment inside these particles. Turns out that that's not the case for the quantum optics aficionados out there. We did anti-bunching studies on these particles, and we can tell you that actually emission is coming from much more than just a few emitters inside any one of these particles. But clearly something at the surface, these transitions are able to efficiently transfer energy to certain loss mechanisms that are killing them. Again, getting more into the kinetics, we see as particles get smaller, their emission lifetimes of all the transitions get shorter and get much shorter. Again, telling us that somehow energy is going to different places than they do in the big particles, and it's of course somehow surface related. So, just to summarize here, if you want to now make particles small, you have to start thinking about different optimization parameters, different constraints. So small UCNPs, there's clearly energy transfer to the surface that's going on. And if you want to use these as single molecule probes, if you want to look at individual ones of them, you always are using higher excitation power than you use if you're imaging ensembles. So knowing these two sort of constraints, that means you're basically dealing with different kinetics or their new design rules for making these particles. So enter simulation. So Emory Chan, who also works at the Molecular Foundry, had been playing with these up-converting nanoparticles for a while, and he was actually very interested in in being able to tune the color of them and the lanthanides and make them spectrally pure and stuff like that. So he started working on building up a model that allowed him to sort of understand all of the different relaxation rates and energy transfer inside these particles. And so he started working on a model that ended up getting a lot more in depth than even he had thought. It takes into account all sorts of different processes in these particles, and in the end actually accounts for all of the hierarchic states, or all of the manifold to manifold transitions that exist for all of the different lanthanides inside these particles. And this, when we heard this, we were like, wait, we can use this model to immediately plug in our new kinetics and start to figure out what's going on in these structures. So we talked to Emory. We immediately, okay, so we first started with what people think. The conventional wisdom is that you take your bulk particles, you put in a little bit of YB, or a lot of YB, a little bit of erbium, and that should be optimal. And he found that that indeed was the case when you're illuminating at what people typically use for doing ensemble imaging experiments, so 10 watts per square centimeter, for example. What you see here is that the optimal nanoparticle has emission is brightest when you have 20% YB and roughly 1 or 2% erbium. When you add more erbium, you start to get this sort of, intersystem kind of relaxation going on, basically self-quenching in the particles. And so that's why no one puts more than 2% erbium in these particles. However, the system gets a lot different when you essentially move up to what are single particle imaging powers. What you start to see is that unlike up here, where there's sort of a maximum in terms of erbium percentage, if you're operating at something like 10 to the 5, 10 to the 6 watts per square centimeter, now you want to shove in as much emitter as possible. And the reason for that is because now you're essentially saturating a lot of different energy levels. Plus, you're competing, particularly for small particles, you have different rates, faster rates that you're competing with to the surfaces. And so now this self-quenching is no longer the dominant loss mechanism, and you can put in as much emitter as you can in these particles. The other thing you notice is that the effect of this sensitizer, the ytterbium, is much less. While, okay, it certainly helps to still put in more ytterbium and have it act as an antenna, it doesn't help nearly as much as it does in sort of the previous case. And in fact, if you now plot sort of luminescence for lots of different powers and as a function of erbium percentage, you see that somewhere around 10 to the 4, 10 to the 5 watts per square centimeter is where you have this sort of turnover in kinetics. And so this means that if you really want to use these particles for single particle imaging and for some other things, you need to account for the kinetics, and you need to, of course, make the particles a totally different way. So we did that. So instead of so here now is an image of a couple of particles that are made the old-fashioned way, 20% terbium, 2% erbium, and then we made some that have no terbium and are 20% erbium. That's sitting right about here. At low powers, only the other ones emit, but as you start to push the power up, you actually see that actually the new particles get much, much brighter. And you can actually see that in this plot here where we're simply graphing the particle luminescence intensity as a function of laser intensity. At low intensities, again, the other particles are better. In fact, these new particles, you never would have discovered them, right? Because they're non-luminescent at ensemble imaging powers. Only at the high powers in these small particles does this really start, sort of this new composition start to shine, pun intended. Okay, so then now we can make smaller and smaller particles. That's what we're showing here. And it turns out that we were now able to make what turns out to be the smallest upconverting nanoparticle that anyone's ever imaged, a sub 5 nanometer particle. You can see that if you make a sub 5 nanometer particle the old-fashioned way, you just can't see it no matter how hard you pump it with your laser, but you can clearly see them, see the new formulations. So these are fantastic. And this got us excited because this means now we have particles that we can make smaller than 30 nanometers. You could use 30 nanometer ones as well, but we can make them small and we can see them. So we thought that was pretty neat. Okay, so the lessons here were to make brighter single molecule imaging probes. Maybe the new approach should be to ignore ensemble properties. First you have to identify where all the energy is going in your system. What are the bottlenecks in terms of losses and kinetics? And then you start to optimize them with those constraints in mind, not pre-screening out a bunch that probably could be good. And so we think that's one of the main contributions of what we learned here. So I say here you want to optimize for higher fluences and environmental constraints, and so that sort of brings me to the second part of my talk. I would count higher fluences actually as an environmental constraint, but the point is that you need to think about where you're putting your particles and then start designing them for these purposes. And so going inside tissue, has a lot of constraints. Right? So can we use upconverters to probe deeper inside your brain? So something that was alluded to by both of our first two speakers. So optical imaging is great, and it can give you high spatial resolution and very fast temporal resolution, especially if you can do this time dilation. The main issue, right, with using photons to try to probe into tissue is that photons don't go very deep. Basically, if you want to image, you need ballistic photons, and so on the order of 100 microns, most of your photons end up getting absorbed or scattered inside of any tissue. And so what that means is that any optical imaging of tissue or your brain all has to happen within, say, the first 100 microns or so. So that's the width of a human hair, right? And so really what you'd like to be able to do is to go deeper. And in fact, with the ultimate goal being to probe the entire structure. So this paper totally changed how we think. So this is maybe old hat to a number of you, but this is a paper that came out about 1.5 years ago, came out of Chris Schu's lab at Cornell, where he showed that using 3-photon imaging, he was able to see more than 1 millimeter into tissue, so more than an order of magnitude deeper than really what people have been seeing. People have pushed to 200 or 300 microns before, including him, but really getting beyond 1 millimeter is a big deal, at least it was to us. So the way that they did this is they realized that as you move to higher and higher wavelengths, okay, your absorption in water goes up. But at longer and longer wavelengths, your scattering goes way down. And so what we're plotting here, the green line in particular, is a plot that is a model that accounts for both absorption and scattering. And you can see that actually there are some other areas. So this is attenuation length. So you want attenuation length to be long in this case. They had previously done studies at 800 nanometers where attenuation length is actually pretty short, and at 1,300 nanometers where it gets a little better. But maybe you could do better by going out to 1,700. And so they did this. Now, of course, most fluorescent dyes and proteins that exist out there, you can't do a 2-photon absorption anymore, so you had to move to 3-photon. And that's what they did. And so here is emission coming from the structure. So they went to 1 millimeter, but what actually limited them wasn't getting the 1,700 nanometer light into your head, or into the rat's head, or in this case. It was actually getting the light back out. So in this case, they were actually limited by the existing dyes and probes that exist at this level. So they were still trying to detect visible and some basically visible light coming out of your head and some near infrared. Of course, they're also limited by the fact that you have to turn your laser power way up, even if it's short pulses, in order to get enough emission, again, when you're 1 millimeter inside the tissue. So, this made us think, well, what if there was a way that was, 5 or 6 orders of magnitude for two-photon for a 2-photon process, maybe even more efficient relative to a 3-photon process for getting this kind of luminescence, right? So this led us to think about maybe upconverters can be ideal for this. So again, as I say, right, we know for 2-photon processes, we're already many orders of magnitude more efficient. Can we use this for 3-photon? It actually turns out that for a completely different reason, we'd done 3-photon imaging of upconverters a couple years ago. We were actually using them as probes of some epsilon near zero metamaterials that were also made at the foundry. Here we were using communication wavelength 1550 and then upconverting to the green and the red transmission from our old nanoparticles. But now with the new ones, can we design them specifically for this case? So we go back to our model, and we start plugging in some parameters. We want a particle that can absorb around 1,700 nanometers and emit around 800 nanometers, something much more friendly for getting in and out of the tissue, not the 500 or 600 nanometers that had been used before. And we came up already with a few different possible compositions for these particles. Most of them involve thulium, and then various amounts of gadolinium or terbium or erbium, and some with holmium. But you can see that actually we can get ones that are relatively bright, and in fact it turns out that it predicts that for the same powers that we typically use for 2-photon upconversion, they're only about an order of magnitude dimmer for 3 photon of conversion. So this is, again, was pretty promising. So where are we with this? Well, we just made our first batch like a few weeks ago. So this is what these particles look like. We're excited to stick them in our system. If any of you are excited to stick them in your system, let us know. We're quite happy to show that these things might, work. So, all of that is okay, great for if you want to get light in and light out, but you don't always need to get light out, right? So, we've been trying to talk people into the fact that you can use these things essentially as little nano light bulbs that can go deeper into your tissue. So we finally started up a project with Udi Issakov's lab. The idea is that, okay, you can use 1700, or actually it turns out even 2100 nanometers. Is also a promising wavelength for these structures. You can get that to go deep into your tissue. Then you can have a little nanocrystal that upconverts that to visible or even UV light, and you can use that light to stimulate something. For example, in this case, I think this is a glutamate sensor from Udi's lab. And so you can turn these channels on and off, for example. So now you can start thinking about doing optogenetics deeper in the material. And just to point out, it's not crazy to think about using these things as light bulbs for the wavelengths that you want. You can definitely get sort of the little UV wavelengths that you want. You can also get 400 nanometers, they're just not shown here, and then really span the whole visible range. The cool thing, too, is that you can actually change the ratio between the UV emission and the visible emission simply by modulating the power of your laser. So this gives you a way to, if you want to switch back and forth, for example, you can do that with the same particle. Of course, you don't always need to do that. Something else that we've been working on and has been getting a lot of attention in the literature as well is using these particles, it's very much related to the last slide, as sort of essentially FRET donors, or luminescent resonant energy transfer donors. So some people call it LRET. We prefer FRET with a PH for phosphorescent energy transfer. But again, the idea being that you excite your particle, again, using friendly infrared light, and then it can transfer its energy efficiently to these other particles. Remember, these particles actually are pretty fantastic at transferring their energy to the surface. Right now, most of that energy that goes to the surface simply turns into losses and ends up heating the solution, for example, or the ligands that are nearby. But if you could replace those with something you actually wanted to accept your energy, that's great. Right? So there you go, transfer your energy to, in this case, these are rhodamine molecules, I think. Another cool thing, and this is sort of the idea that got us excited about FRET with a PH, is remember I told you most of these energy levels, for example, in erbium or thulium, have relatively long emission, long lifetimes, typically in the order of hundreds of microseconds. And that's another fundamental limitation to the brightness. You can only turn your emitter over every 100 microseconds or so. On the other hand, a lot of these other emitters that have allowed transitions have lifetimes on the order of nanoseconds. And so by strongly coupling an emitter, an acceptor to the nanoparticle donor, you can actually speed this process up significantly. In principle, making them much, much brighter if all of your energy, even if some small percentage of your energy goes the right direction. And so we're starting to model this now. We think actually we do have ways to steer our energy into the right direction. So this is great, right? Because besides just being brighter, right? I can tell you already, so for a 20 nanometer UCMP, we typically get out about 10 to the 6 counts per second on our APD, which means that we can get about a nanometer's worth of localization if you're doing sort of super resolution type of imaging with these structures. If you can get 1,000 times more, now you're getting, right, nanometers type localization at millisecond time scales. And so it begins to open the possibility of again, seeing real dynamics in these structures, or at least relevant dynamics. And of course, actually, again, being able to couple to these types of other emitters gives you now the possibility for some kind of sensitivity, right? This is a sensor workshop after all. Sensitivity to what's happening locally. So you can imagine having sort of a FRET acceptor that's sensitive to ions, to sodium, to calcium, right? You can imagine maybe something like this being one of Adam's caviar molecules, caviar for calcium, or caviar with a K for potassium, uncaging, sort of uncaging the molecule, or even, leave it at that. So, and again, also maybe gives you the possibility of doing real functional sensing. So that's, and I think, anyway, so I think there's a lot of promise in these areas. It's all on it's all on PowerPoint right now and in proposals, but we think that there's a lot of possibility here, and so this is why I'm excited to be down here. We'd like to find ways to make this happen, to test these out, decide if these ideas are total bunk or if they're good, by talking to people and sort of accelerate that whole process, that turnaround process. Again, looking forward to talking to people. So, okay, just looking forward to the future, right? Things to point out. So this is really still sort of a young technology as far as probes and sensors go. However, the community's catching on. Here are just some stats from Web of Science. This is showing you the number of publications about upconverting nanoparticles that have been happening in the last few years. It's clearly ramping up. Again, other issues still continue to be chemical functionalization, specific targeting, and even delivery. I'd add to that same issues as that Alan mentioned. I didn't know about the ultrasound methods for pushing things past the blood-brain barrier, so that actually is very promising for these particles as well. But really, I think a lot of chemical and functionalization and targeting has to be worked out for these. I mean, some of it's not going to be that different from quantum dots, but others is going to be. And of course, it should go without saying, we need to find ways to make them better and better so that we can work at lower optical power. So I was talking about powers of 10 to the 6 watts per square centimeter, which, I don't want that in my head. But actually, we already think that getting to 10 to the 4 watts per square centimeter isn't going to be too difficult. Remember, the 10 to the 6 watts per square centimeter was for seeing a single 5 nanometer particle. But you don't always have to see a single particle. And we're certainly not going to start there. But also, we think we have ways of playing with very different illumination schemes where we can drop the power needed by a couple orders of magnitude. And we're also working on techniques where we couple our particles to antennas, essentially, that allow us to use much lower intensities and then just create very high intensity locally. So that's why we're dealing with that. So to conclude, I think hopefully I've convinced you that at least in certain ways, UCNPs are ideal single molecule probes. I think people should start rethinking how they approach the design of some of their emitters. In particular, I think there's probably a large class of molecules that have been discarded as probably being bad probes, but maybe aren't. And finally, we think that there's a lot of potential use for these types of things and related nanoparticles, things that are nonlinear nanocrystals that can give you third harmonic generation, for example, in terms of deep tissue imaging. Before I finish, let me, of course, thank the people that did really most of the work here. I already mentioned Emery Chan and Bruce Cohen, who are my prime collaborators on this project. Dan Gargas was the guy who did most of the photophysical studies, a postdoc of mine who's now moved on to Seagate. And of course, Andrew again for making our pictures. And thank you for listening.
Hi, are these are all paramagnetic, or can you make them paramagnetic?
Yeah. So the question is whether these particles are paramagnetic, or can they be? And this touches on a discussion we were just having a break. So I think the answer is yes. So so first I should say I don't know for sure. However, we make a fair amount of our particles with a lot of gadolinium in them, which is. And so that can be doped in at pretty much any level you want, at least up to 20%, probably higher. And so I think possibly, and I haven't, to be honest, we haven't spent a lot of time thinking about the magnetic properties of a lot of the other lanthanides. I know, I'm sure other people have, but so I think there is real promise there.
Base. Is this okay? A very fundamental question, but how do you make them bio-friendly? what sort of coating do you?
Absolutely.
Coatings have been developed.
So there's, again, most of the stuff that's in the literature has very much been duplicating what people do for quantum dots, as far as I can tell. We, again, we haven't done a ton of bioimaging. We're just putting them in now. We just usually put an amphiphilic polymer around them, and that gets them into water. But that's about as specific as we've gotten ourselves. Now there is a fair amount of literature on these for targeting, but that's what we've been doing right now.
What sort of thickness do you need to protect it?
So
Prevent aggregation, et cetera.
I don't have a good answer for that. Again, all my answer would rely back to what people do for the nanocrystals, and I can't think of a reason why it would be much different for these, but I think that's exactly right. The nice thing is that you don't have to necessarily add an extra passivating layer to make them not blink or something like that.
Is there any physical property that can be voltage sensitive or electric field sensitive? Just following the first talk today.
Not that we know of, but I also think we haven't thought hard enough about it. Again, right now our strategy is to more have it coupled to something that is voltage sensitive or chemically sensitive. Again, we think there's a lot of room for design here because clearly a lot of energy is going to the surface for these particles. I mean, I think they are again, to the extent that they're somewhat sensitive to changes in index of refraction around them and stuff. But that's nothing that's useful in these applications. I think you'd like to tie them to something that's more sensitive. At least that's been how we're thinking about it. I'd love to be wrong.
Thanks. So, I'm on? Okay, great. So the electronic spectrum of these things looks a lot like a barcode, and obviously one of the nifty things about these is that they've got actually a bunch of different transitions that you can excite in order to bring them up into their optically active state. Can you use that in some way to multiplex them? I mean,
absolutely. In fact, we wrote a paper about that a couple years ago on it. Exactly. I mean, I think that you can multiplex both the emission and the excitation in principle for that reason with relatively small shifts in wavelengths. So absolutely, I think that's a real plus for these things. I mean, so you're not tuning their luminescence by changing their size like in a quantum dot, but you absolutely get these different emissions. And so you can kind of go both ways. You can actually purposely have lots of emission lines that we often see here, or you can engineer them. This is essentially why that model was built, so that you can just favor really one of the transitions and get sort of spectral purity. But I think both ways work, and in fact, I think the multi-lines, in the end, there's more information there.
Can you comment again on the up conversion efficiency of these lanthanide-based nanoparticles? And also, have you thought of using them in photovoltaic, for instance, to recycle infrared photons?
Can you repeat the first part?
I just wanted you to remind us again the up conversion efficiency
right.
of the lanthanide-based system. And
I can't remind. I purposely didn't tell you. So they're not particular. I mean, so this is an up-conversion process, right? It's a multi-photon process. It appears, so you will find answers, you'll find that number in the literature. That number varies by orders of magnitude depending on what paper you look at. We tend to think that, so it gets better, it turns out, as you turn your power up because you get to saturate sort of this intermediate level. On the order of 0.1%, I think, is a fair number for these particles. But it's a two-photon process, right? Sort of. You do start to saturate, and it gets more and more linear in behavior. Are people using these for PVs? Absolutely. In fact, some of our users at the Foundry, something I didn't mention, the Foundry is also a user facility like a synchrotron. People write proposals, and then they can come work with us for free. So people absolutely are using these for photovoltaics. I will save my comments about whether that's actually useful for after the session. But But yes, that's, the idea is a good one. Why not use the IR light that you're throwing away anyway to at least get a few more visible photons out? The question is how much does it perturb the system already once you do that? But it's good. I mean, in principle, it's a good idea.
Thank you. I have two questions. The first is if you're gonna put a passive layer on the surface and lower the excitation power density, can you still achieve the same like luminescence brightness?
So by adding the passivation layer,
And lower the excitation power?
can you still get the same bright? Great question. So I think you do have to be smart about putting on your passivation layer because that is a way that there's this, well, for a number of reasons, but also because there are specific bonds and ligands that you could immediately transfer energy to that could, also, again, change the kinetics and kill your sort of your energy branching. But that said, I think also there's still plenty of room for improvement in terms of making them even brighter. I think I've showed you all the compositions percentages that we've tried. There's a lot of space to play around in the middle. I mean, that's more of an optimization problem, and so we have a robot working on that. But I do think that actually you can improve the brightness more. And we know, so it turns out one of the cool things about the study that I didn't mention too much is that for these particles, it looks like they're very good. There's effectively an energy migration length that's about 2 nanometers, sort of that we discovered through 2 different independent paths. So that's basically what you need in order to get some decent passivation on this structure. You actually get energy migration for much further, but the main stuff happens within a couple of nanometers. I think this maybe gives you some information about fret distances and things like that as well.
Okay, thanks. So the second is if the size of a particle is comparable to biomolecules like 4.8 nanometer, and how do you control the stoichiochemistry between like a biomolecule and a nanoparticle and to make single molecule imaging?
I'm hoping you could answer. No. It's a great question. It's a great question. That's, I think, really a lot of this. There's not you have to pick the right functionalization, and that opens up. Then that alludes to your previous questions.
Thanks.
This may be jumping the gun a little bit, but do you know anything about how these things diffuse, either just in liquid or in tissue, like in a brain? And have you ever just tried giving it to an animal to see if they live till the next day?
So actually, yeah. So there have been studies on this. I'd say to summarize, I'd say the diffusion for the ones that are, that have like an amphiphilic polymer on them or something tends to look very similar to what quantum dots do. The animals do seem to live, and I'm only familiar with maybe 1 or 2 toxicity studies on these particles in particular, and the short answer there was that they appear to be less toxic than quantum dots by about an order of magnitude. I don't know what that order of magnitude is. There was a 10x, but I think in terms of concentrations.
Have you guys studied their down-converting properties of your small probes?
Not a lot. Right. So you can certainly use these for down conversion, and actually they're relatively efficient for that kind of thing.
Especially in the 1700, like you see in like this region.
Absolutely. In fact, right now, so this is actually something we're just starting to do. In fact, I suspect that in our bigger particles, or much bigger particles, we're actually getting random lasing inside these things because we're pumping them so hard, and in fact. And we can see interesting changes in luminescence intensity as we turn our power up and up and up. But we haven't looked at it, mostly because we're just using silicon detectors. So we haven't spent a lot of time out looking at 1550 or 1700. No. So I think there's other people have. I mean, certainly a lot of these studies grew out of people making things like erbium lasers and YAG lasers and stuff like that. So there is a fair amount of literature on some of these, but in the nanoparticle sense, we haven't spent a lot of time looking at that.
Thanks.
Well, thank you very much, Jim. And so with that, we can go to our lunch break. We're a little early. The food may or may not already be there, but we have an hour and a half. So we can return at 1:30 for an afternoon to expose you to 3 yet different areas for how to approach this, which will be presided over by Nick Spitzer.